Okay, thank you. All right. So an animal usually starts out as a one-celled zygote, and the majority of these zygotes are formed by merging of two haploid gametes, which are sperm and ova, and that turns into a diploid zygote. There are a few exceptions to this rule because sponges, the most simple animals, they can reproduce asexually as well. And some rotifer species are entirely female and reproduce via unfertilized eggs, which is pretty strange. And um, and after fertilization occurs in most animals, the original cell goes through simple mitosis, which is also called cleavage, and eventually becomes a ball of cells called a morula. And the beginning zygote and all the cells up to the morula point are totipotent, which means they can differentiate into literally any type of cell based on the environment. And the cells of the morula later divide a bit further, and then they begin to form like a hole in the middle that's filled with a fluid called blastocele, and it forms something called a blastula. And it's like kind of like a ball with an outer layer of embryonic cells, and in the middle you have the blastocele fluid. But in mammals, it's a little bit different. It forms a blastocyst instead. And the blastocyst consists of a trophoblast, which is an outer cell layer, and an embryoblast, which is the inner cell mass. And the inner cell mass develops into the actual embryo, while the trophoblast develops into like the support organs, like a placenta. And the inner cell mass is pluripotent, and pluripotency means it can differentiate into any type of cell except for support organ cells like a placenta. And if you ever heard of like embryonic stem cell research, those are the type of cell that they use for those are the type of cells that they use. And up until the blastula stage, there are no major differences between animals, but there is one tiny difference in the way cleavage occurs, which I will talk about later because it's pretty important later on. And now we have probably the most important process in all of animal development, which is called gastrulation. And what gastrulation does is it develops a digestive tract. And the reason it's so significant because it's also the first instance of divergence between different animal groups. And it occurs in every animal except for sponges of the phylum Peripherum. And basically what happens in gastrulation is a part of the outer blastula layer folds into the blastocele area. And this folding is called invagination. And it folds at a certain place called a blastopore. And the blastopore, the hole, it forms like the digestive tract. And this fold can either stop midway or it can go all the way to the other side to create a full digestive tract at two ends, which depends on the organism's symmetry. Gastrulation's completion results in the embryo being divided into different layers called germ layers surrounding the digestive tract. And every animal that undergoes gastrulation has at least two germ layers called the ectoderm and the endoderm. But animals that form a full digestive tract form a third layer in the middle called the mesoderm. Now you may be wondering why, out of all of the things to develop, the process that forms the digestive tract is the most important process in embryonic development. Well, that's because it forms like a sort of body plan for the embryo. And by arranging cells into germ layers, it creates this body plan which eventually allows differentiation and forming of different types of tissues. And ectoderms normally form into epithelial and nervous kind of systems. And the endoderm develops into like the digestive and respiratory systems. And for animals that do have a mesoderm, it develops into like a muscle and circulatory systems. And mesoderm forming animals can develop a body cavity, which is called a coelom. And the coelom surrounds the main digestive tract and separates it from other organ systems so that stuff from the other organ systems doesn't just randomly flow into the digestive tract. And this is also where a difference in the earlier cleavage can lead to the blastopore forming either the mouth or the anus. And now I'm going to talk about the differences caused by gastrulation. And first of all, sponges in the phylum Porifera, as I said before, don't undergo gastrulation. They stay as a blastula. And because they stay as a blastula, they don't have any germ layers, which means they don't have any specific differentiation, which means they don't have any true tissues. If you've ever seen a sponge, they kind of just sit there and just absorb nutrients through like regular diffusion. And a slightly more complex phylum is Cnidaria, which is like jellyfish, who are radially symmetrical, meaning like if you divide them at like any angle, it'll have two separate halves. And they undergo partial gastrulation. And what that means is the invagination stops midway. It doesn't go all the way to the very end of the embryo. And because of that, they only have one digestive opening, which just sounds weird because you're excreting waste and ingesting nutrients from the exact same hole. And Cnidarians only have an ectoderm and an endoderm as well. They don't have a mesoderm. So that deeply restricts their complexity compared to the other phyla, which have bilateral symmetry. Bilateral symmetry, basically, if you cut a bilateral animal in half at one specific place, it'll form two equal halves. Like if you cut a human in half, which just sounds wrong, it'll form two equal halves. And bilateral animals with bilateral symmetry, the fold extends all the way to the end of the embryo and forms a mesoderm as well. And the differences in bilateral animals usually come to whether a coelom exists and what the blastopore becomes. And flatworms, or a phylum called Platyhelminthes, have no coelom. They straight up have no coelom. And nematodes, which are also called roundworms, and rotifers have a partial coelom that overlaps with the endoderm. The five main classes of true coelomates, with the coelom entirely in the mesoderm, have even more divisions based on their initial cleavage. And the type of cleavage determines if the blastopore becomes the mouth or the anus. And if you've ever heard of protostomes and deuterostomes, this is what they are. Protostomes undergo spiral cleavage, and the blastopore becomes the mouth. And the other opening that forms at the other side becomes the anus. And deuterostomes undergo radial cleavage. The blastopore becomes the anus and the other end the mouth. Protostomes include mollusks, annelids, which are also known as earthworms, and arthropods, while deuterostomes include echinoderms and chordates. We're chordates, so our butt actually forms first. And here's like a sort of simplified evolutionary tree of what's going on. First, you have the sponges, which have no true tissues, and then it sort of evolves a little bit further. You got radial symmetry for Cnidaria, and then you got even more complicated with bilateral symmetry, starting with no coeloms, and then stuff with like a kind of partial coelom, and then it expands to people who have a full coelom, and then it divides based on cleavage. And if you're confused on what a coelom is or what radial and spiral cleavage is, these are some diagrams as to what's going on. And yeah, this is how like an this is how we are arranged. BU coelomata. We have our endoderm in the middle, and we have an ectoderm on the side. Our ectoderm develops into like our skin and our nervous system and stuff. And the endoderm develops into a digestive tract, and our coelom is like fully inside the mesoderm. And for radial cleavage and spiral cleavage, it's kind of weird. For radial cleavage, when the cleavage occurs, all the cells are perfectly aligned in like an 8-cell mass. While in spiral cleavage, they're not exactly aligned. They're like at an angle. There's like a certain lump of cells at one side, and then everything else is in one plane. And thank you.